Good morning, everybody. This is Ramba. Warm welcome to Success Journey TV channel. Today we are going to discuss about an experiment that is Mohl's of leaf experiment. So this experiment is very important in photosynthesis. This experiment proves importance of carbon dioxide or necessary of carbon dioxide in photosynthesis. So the aim of this experiment is to prove that carbon dioxide is essential or necessary for photosynthesis to occur. So carbon dioxide is a very important factor for photosynthesis. If the carbon dioxide is available in plants, the photosynthesis process will take place and starch is synthesized. If the carbon dioxide is not available, the plants did not synthesize the starch. So we prove the experiment of Mohl's of leaf experiment, the given below procedure. The material requirement of this experiment is a healthy potted plant, usually with well grown leaves. Next one, wide mouthed transparent glass bottle, one cork fitting the mouth of the bottle with a split hole in the middle. Next, potassium hydroxide KOH solution or pellets. This absorb the carbon dioxide in the transparent glass bottle. So this is a very important bit. Which chemical is used to take in transparent glass bottle in Mohl's of leaf experiment? So the KOH potassium hydroxide solution or pellets we can take in a transparent glass bottle. This absorb the carbon dioxide inside the transparent glass bottle. Next, water and iodine solution to test for the presence of starch, and beaker, a test tube, spirit, alcohol, water bath. So these are materials required for the Mohl's of leaf experiment. The materials which are used in test for the presence of starch, that materials also used in this experiment. So next, procedure. Preparing the plant. Place a healthy potted plant in a dark room for 2-3 days for destarching. This completely remove any storage starch from the leaves. So before conduct the experiment, we keep the potted plant in a dark room, because when we keep the potted plant in a dark room, the photosynthesis process will not take place. So the plant consume the starch which is already present in leaves. So after 2-3 days, the completely starch which is present in the leaves, that is consumed by the plant. So this process is called destarching. Next, setting up potassium hydroxide. Take some potassium hydroxide solution in a wide mouthed transparent glass bottle. Arrangement of leaf. Select any one of the leaf from the destarched plant. Carefully insert the leaf through a split cork, ensuring that midrib is not damaged. In such a way, we arrange the leaf in such a way that half part of the leaf is inside the transparent glass bottle and the remaining part of the leaf is outside the glass bottle. Leaf in bottle. Ensure that the half of the leaf is inside the bottle above the KOH solution. The remaining half of the leaf will be outside the cork in the open air. Next, sealing tightly. Surely seal the cork to the mouth of the bottle, ensuring that the air inside the bottle does not mix with the outside air. The KOH solution inside the bottle will absorb the carbon dioxide from the air present inside the bottle. So the appliances should be airtight by applying Vaseline or grease at the mouth of the transparent glass bottle and vertical split cork. The KOH solution inside the bottle will absorb the carbon dioxide from the air present inside the bottle. So this is the importance of potassium hydroxide solution for we taking in a transparent glass bottle. Next, placing in sunlight. Placing this entire setup in a sunlight for 4 or 5 hours, photosynthesis will occur during this time. So when we keep the entire arrangement in bright sunlight nearly 4 or 5 hours, the photosynthesis process will take place. Next, to collecting the leaf. Remove the setup from the sunlight and detach the leaf from the plant. So we remove the leaf from the potted plant and also remove from the transparent glass bottle and allow the leaf test for the presence of starch. First, next to starch test. First, dip the leaf in hot water to soften it. So in a starch experiment, first we take the leaf which we insert in the transparent glass bottle. So that leaf is boiled in a hot water. Next, place the leaf in a test tube with alcohol or spirit and heat it in a water bath. This removes the chlorophyll. So this leaf, after boiling the leaf in a beaker, then insert the leaf into the test tube with containing alcohol, methylated spirit, and boil the test tube along with water and methylated spirit in a water bath. After 4 or 5 minutes, the chlorophyll which is present in leaf, that is completely removed from the leaf and enter into the methylated spirit from the leaf, causing it to turn pale white. So when we boil 4 or 5 minutes, the leaf completely turn into pale white color from green color. Finally, remove the leaf and wash it with the cold water. Place it in a petri dish and add a few drops of iodine solution on it. So finally, we remove the leaf from the test tube and keep it in a petri dish and add 2-3 drops of iodine on the surface of the leaf and observe the color of the leaf. Next, to observation and result. The part of the leaf inside the bottle, this part will not turn in blue black with iodine solution. This indicates the absence of starch. The part of the leaf inside the transparent glass bottle, that part is not turns to bluish black color because there is no carbon dioxide available in a transparent glass bottle. So the photosynthesis process will not take place and starch is not synthesized. So when we test the iodine, with iodine, the leaf is not turns to bluish black color. Next, to the part of the leaf outside the bottle, this part will turn blue black with iodine solution. This indicates the presence of starch. Next, the outside of the part, when we observe, it receives the carbon dioxide from atmosphere. So the photosynthesis process will take place outside the transparent glass bottle. So in this part, when we add the iodine solution, it turns to bluish black color. Why? Because this portion receives the carbon dioxide from atmosphere and carries the photosynthesis process and prepare the starch. So this starch mixes with iodine and turns to bluish black color. So finally, conclusion. The part of the leaf inside the bottle did not produce starch because the KOH solution absorbed the carbon dioxide from the air inside the bottle. So the part of the leaf which is inside the bottle, that is not turns to bluish black color because the carbon dioxide which is present inside the transparent glass bottle, that entire carbon dioxide is absorbed by the potassium hydroxide solution. Due to the lack of carbon dioxide, the photosynthesis did not occur. The part of the leaf outside the bottle produced the starch because it had access the carbon dioxide. Therefore, this experiment proves that carbon dioxide is essential for photosynthesis. So the part of the leaf which is outside the transparent glass bottle, that receives the carbon dioxide from atmosphere and carries the photosynthesis process and prepare starch. Precautions. This is very important precautions for Mohl'S leaf experiment. So first one is destarching. It is crucial to place the plant in the dark for 2-3 days before starting the experiment, so that the starch in the leaves is completely removed. So before doing this experiment, the destarching process is a very very important process. In this process, um, uh, in this process the potted plant, when we keep the dark room nearly 2-3 days, the entire starch which is present in the leaf, that is completely consumed by the plant, and also there is no photosynthesis process will takes place due to the unavailability of sunlight. So next process, tightening tight sealing. So this process is also important for one of the precaution, that is ensure that the bottle mouth is tightly sealed with the cork, so that the outside air does not enter into the bottle. So the sealing of the transparent glass bottle is a very very important process. In this process, the mouth of the transparent glass bottle and vertical split cork where they connected, that place is sealed with Vaseline or grease very tightly to cut the air, connection from outer atmosphere to transparent glass bottle. Next precaution, caution with KOH. Potassium hydroxide is a strong base and harmful to the skin. Handle it carefully and avoid directly contact with hands. So when we pour the potassium hydroxide, we should proper care um, while pouring the KOH solution into the transparent glass bottle. Next, no damage to leaf. This is also an important precaution. Ensure that the no damage occurs to the leaf when inserting through the cork. So insert the leaf through the vertical split cork properly without damage of the leaf. Next, to time. This is also important factor. The plant should be kept in sunlight for at least 4 to 6 hours for sufficient photosynthesis to occur. So the time when we keep the potted plant below the less than 2-3 hours, the photosynthesis process will not take place. So the result, we cannot get the accurate result. So for that, maximum 4 to 6 hours is essential for accurate result. Next, heating alcohol. Alcohol or spirit should not be heated directly over a flame, as it is a flammable. Always use a water bath. So we should not directly boil the methylated spirit uh, in the test tube. So why? Because it is very flammable, so it catch the fire. So we boil the methylated spirit and leaf in water bath, but not directly on a flame. So this is also important instruction. Next, iodine solution. Ensure that the iodine solution is fresh and capable of turning blue black in the presence of starch. So we don't use any expiry iodine solution. We should take fresh iodine solution for a better result. So this is the important experiment for photosynthesis of Mohl's off leaf experiment. So please like, share and comment and subscribe channel. Thank you for watching.